There are lots of different kinds of nodules, and much of the pioneering work on their shape was done by a gentleman called H.D.L. Corby, commonly known as Tom, in what was then Rhodesia, is now Zimbabwe. And over about 10 years, Tom Corby collected nodules from there and from other parts of the world, and in 1980 he published a thesis with some lovely diagrams in, all of which he drew himself. Of course, this was long before we had word processors. And when he retired, he gave me his collection of nodules, and I have them in these cabinets here. We do have 19,000 species of legumes, not all of which can fix nitrogen. In other words, not all nodules are equally effective.
This tree is a legume, a member of the Leguminosea. The Leguminosea has 19,250 species, divided into 3 subfamilies. I've got the flowers of this tree in my hand here, and you can see they're beautiful yellow flowers, but they don't look like pea flowers. Now, this family is called, this subfamily of the Leguminosea is called the Caesalpinioideae, difficult name. But the reason that that's important is that many trees in the Caesalpinioideae, like this one, don't actually form nodules. They cannot nodulate and fix nitrogen. Now, all legumes have nitrogen-rich leaves, and it was thought that nitrogen fixation evolved as a strategy to satisfy their nitrogen demand. But because this family is really the oldest legume type in terms of evolution, it shows that nitrogen-rich leaves came before nodulation. So why would legumes have nitrogen-rich leaves? Well, basically, the more nitrogen in the leaf, the more rubisco, that's the enzyme for photosynthesis that you have, and the more carbon you can fix from the atmosphere in photosynthesis. So they evolved to have nitrogen-rich leaves, and then some of the legumes later evolved to be able to nodulate and fix nitrogen in order to satisfy that demand. Now, above and behind me, there's another legume tree I want to show you. These are its flowers. You can see they're very different. This is a little ball of white stamens, actually. It's the male part of the flower is all that you can see. And this species is called Leukina leucocephala. Leuco meaning white, creamy, cephala meaning head. And you see it's got little white heads. This is a member of the Mimosoideae. Many of you might know mimosas as yellow flowers, or the acacias, which are members of that family, subfamily. These nearly all nodulate and fix nitrogen. So we've got 3 subfamilies, the Caesalpinioideae that I just showed you, the yellow flower. This is the Mimosoideae, and the one you're all really familiar with are the pea-type flowers, to which all of the useful crop legumes belong.
So this is a pea plant that we've been growing in a special medium. We're growing it in agar, and the reason we're growing it that way is so that we can see the nodules that form after we add the Rhizobium bacteria to induce nodule formation. And so we could, for example, just take some soil and inoculate bacteria from the soil and put it onto a plant like this, and we'd get nodules. And what we can then do is take these nodules, either from this plant here or even a plant in the soil, and cut out the nodules and crush the nodules and then spread the bacteria onto what we call an agar plate so that we can see them after a few days. We see the bacteria growing on the plate. And so this is how we get the bacterium that's called Rhizobium leguminosarum out of these nodules, which are the pea nodules. And what we did some years ago was we took a strain of Rhizobium leguminosarum and used what some of you will know is called a transposon, or if you like, a jumping gene, and we used that to make mutations. And the advantage of that is once that transposon is in a specific gene, we can find that gene. And so what we did was made lots of mutations and screened those mutants to find some mutants that wouldn't form nodules on the plant like this. And those bacteria were unable to form nodules, so we knew we had mutated part of the signaling between the bacterium and the plant. And so what we then did was identified the DNA that corresponds to the genes we had mutated, and we linked that DNA up to what we call a reporter gene. And that tells us if the genes are on or they're off. And we found that when we grew the bacteria in lab culture, the genes were off, which meant that it suggested to us that in order to switch them on, we might need to get them to interact with the plant in some way. So when we put the bacteria onto plants, we saw the genes were on. And so in this plate here, we're illustrating the switch on of the genes, because what we've done is put a lawn of the bacteria onto the plate. And then on top of that lawn of bacteria, we've put on little seedlings of a legume, and that legume is switching the bacterial genes on, and you can see that the genes are on because the reporter gene that we've used has switched on and caused this colour to appear, this blue colour, which tells us that these legume plants are switching the bacterial genes on. And so that is the beginning of the communication between the plant and the bacterium. What's happening is the bacterium is recognizing that the plant is exuding signals or compounds, they're called flavonoids, and those signals induce expression of the nodulation genes that are required for the bacteria to communicate with the plant.
It's a process that is highly controlled by the plant, and so what it requires are two major processes. And that's what we will show you here. That's, on one hand, this is a structure, a tube, an infection thread that is made by the plant, and this is guiding the bacteria inside. The second process, it's a very smart process. Here you see that rhizobia induce cell division. And you can say, well, that's just cell division, but it's a differentiated cell that becomes now a stem cell able to form all the tissues of the nodule. So the infection that we have, the bacteria is at the outside and it has to enter the plant by this infection thread. But of course, that's starting at the outside. The cells that are infected are the root hairs. And that's, it are very few bacteria that you need to form a nodule. In fact, one is sufficient. And that's what you see in green here, a single bacterium that you see. This bacterium has landed on the root hair. It starts to communicate with the plant. It sends out signals. And the result of this communication is that it redirects the growth of the root hair. It's no longer this way, but it starts to turn now. So it starts to turn, by which it makes a 180 degrees turn around the bacterium. Well, here you see the result of this 180 degrees turn. It's a nice curl that is formed, and the bacteria are inside. Here it has already done more, because also a tube has now been formed by which the bacteria are going inside. But why would it do so complicated? Why would it first make a curl? Well, that's, I can tell you better in the next slide, because now you see a section through here. That's this here. So you look at the top of a root hair and you see the bacteria sitting inside. Well, if it would have been here, so if it would have done simple, and it would start to form this tube here, it would weaken, remove the cell wall here, and what would be the result? The root hair would explode. The pressure on a root hair is more than the pressure on a tire. That's why it has made this curl, because now it has sealed itself here. This here is a closed environment that you have, a closed environment that is not in contact with the outside, so it can easily weaken a cell wall that you don't see here, because we use a dye at the outside, add it here, and it didn't even reach it. Really telling this is a very closed environment, and that's why it's now safe to start the tube at this place here. So here again we go back to the tube by which the bacteria enter. I explained to you the part here where it enters the root hairs, but in fact it has now to move this whole way. This is the tube, the infection thread that is made by the plant. And another picture that you see, that's and now it's turned 90 degrees, but now it's moving in this way, and it's stained in a different way, but it shows how fascinating these tubes are and grow inward. It looks like a very fancy system. It is, and you might think it is legume specific. Well, in fact, we know now it's not. It is in fact using the same genes, the same machinery that is used in another interaction, and that's the mycorrhizal interaction. That's an interaction in which a fungus enters the plant and forms these nice structures, in this case for the uptake of phosphate. This is an interaction that almost all plants can do. So it's a very ancient system. These plants already had it before they even had roots in evolution. So very ancient and common to all plants. So the root nodule. This is the outside, but how does it look inside? Let's see. Here you see a section through this nodule. And what is nice in this type of nodules is that you have infection taking place here. And in this way it's getting older. And these nice blue cells, that are the cells where the nitrogen fixation takes place. But first, a look up here. Here you see an infection thread, the tube again. It is entering the cells now, and some bacteria have already released, and they are now dividing again. That's what's happening in the apical part of the nodule. That's what's happening here. Then the bacteria, they amplify and they fully fill cells, and that's what we see here. Here you see it's completely packed with nice big rods. That are the structures here. These are, in fact, organelles, nitrogen fixing organelles. It's not just simply a bacterium that has entered, it is a bacterium that is now hosted in compartments of the plant. Every bacterium here is in a small membrane compartment that is made by the plant. And if you label only the host membrane, so that's around it, then you have the next picture. You see here, it's all these are small compartments made by the host. That's where nitrogen fixation takes place. That's where the nitrogenase is doing its job. This is requiring a lot of energy. And it also is an enzyme which is very sensitive to oxygen, a dilemma in the nodule. And in fact, to solve that, in between here, there is still a lot of plant protein. And the most important protein that is doing that job, taking care that oxygen is low, but transported to the bacterium, that's the hemoglobin that is there. And that's what you can even see at the outside. The nice red color that you see here, that's caused by hemoglobin. More than 10% of the protein of the plant, taking care that oxygen is transported towards the bacteria. So the molecule that the bacterium makes that is sufficient to induce most of these steps, or at least essential for all the steps that are described, that's the NOD factor. You see here, a lipid and a small chitin backbone. That molecule does all the jobs. Infection, curling of root hairs, cortical cell divisions, and even a nodule is made of it. And that's what will be illustrated here. What you see here is the surface of a root, and you see root hairs. And now what will happen is we will put a small droplet on one of the hairs, and this droplet will contain one molecule of NOD factor. Then you will see what happens. The plant, the root hair, remembers where the NOD factor is placed, and it will grow in the direction where the NOD factor is sitting. Here you see the pipette. It has delivered the molecule of NOD factor. Root hairs are growing. They all grow straight, except the one that received this NOD factor. It is now growing into the direction where the NOD factor was placed.
We've injected a dye into a single cell on the root. Now that cell's called a root hair cell, and you can see the cell here, which has got this green colour in it, and we've outlined the cell so that this outline here shows you the bit of the cell that we're actually imaging. And what we're able to do is measure changes in calcium. And we think that that calcium spiking is the signal that's being transmitted between the bacteria and the plant cell. We can actually start to see when we add the nodulation factor, that's the one that the bacteria make, the rhizobia, we see changes in pattern of the calcium, and you can see it's going up and down with a regularity here. So the signal that's being formed here is telling the plant cell that it should develop. And that development is inducing changes in gene expression and starting the process of building a nodule.
So legumes, such as this plant Medicago truncatula, evolved the capability to recognize rhizobial bacteria or nitrogen-fixing bacteria by modifying a pre-existing signaling pathway that was required for the mycorrhizal association. That mycorrhizal signaling pathway is very ancient. It was there right at the onset of the evolution of early land plants. And as a result, it's not only present in legumes, but it's also present in pretty well all plant species that we're familiar with, including fir trees, but also importantly, cereal crops. Now, that means the signaling pathway that legumes have utilized in the evolution of nodulation is already there in the important cereal crops, which is where we want to engineer nitrogen fixation. And that's a real silver bullet that will deliver nitrogen without the economic and environmental costs that we associate with fertilizers. But that's a very long-term aim. I would say we're talking 20, 30, 40, maybe even 50 years before we can deliver nitrogen-fixing cereals. And in the meantime, in the shorter term, we still have to improve nitrogen, increase nitrogen in our agricultural systems and do that in a more sustainable fashion.